So the ENCODE project has delivered a wealth of information on the human genome. The genome is the set of instructions that define the biological features of living organisms. In the human, the color of the eyes, of the skin, of the hair, the height, etc., etc. The unfolding of these instructions is triggered by the transcription of DNA to RNA and the post-processing to this RNA to functional RNA molecules or proteins. And from here, all the network of actions that give rise to a living being are orchestrated. So the ENCODE project has tried to define the regions in the human genome that are actually responsible of this biological encoding of information.
In a nutshell, what has the ENCODE project delivered?
So we are part of the ENCODE project since the pilot phase of this project, which started in 2003. And we had always been working on actually characterizing the transcriptional activity of the genome. So the regions in the genome that code for genes and for transcripts, which are the basic units of information.
So what is going on in the human genome?
Well, there is a lot of activity in the human genome, much more activity than we had previously anticipated. For instance, we know that almost 70 or 80% of the human genome is transcribed into RNA molecules that are post-processed. And then there is a lot of regulatory activity that can be measured by binding of proteins to the DNA, by specific modifications of the epigenetic, which is called epigenetic modifications of the DNA, which do not change the nucleotide sequence of the DNA, but change the way in which the DNA is structured in three-dimensional space.
What do these results mean for biomedical research?
Well, I mean, actually they are quite important, although they are very basic. So the better we know how a system works, the better we can act on the system when it does not work as we want. For instance, the better that we know how the human body works, the better we can fight against disease. And we know that a large fraction of the characteristics of the human body are encoded into the genome, into the sequence of the genome. Therefore, if we know what's going on the sequence of the genome, we know better how we work, and therefore we are able to actually develop better strategies to fight disease.
What makes the ENCODE project special?
So I think that one of the things that make this project unique is that the fact that it's using pioneering technologies that are at the frontier of being developed and benchmarked, and also, of course, the fact that it is a project that involves the participation of many groups in a way of doing science which is much more cooperative than the traditional way of researchers working on isolation on their labs.
What lessons have you taken away from this project?
Well, the ENCODE project has actually produced lessons at many different levels. At the scientific level, we now know much more about how the genome works and how complex is actually the working of the genome. And finally, there are the lessons in sociology of science, if you wish, in which we have and we still are learning how to work on large consortiums, on scientists working in all across the world with many time zones differences, which makes direct communication sometimes quite complex.
What's next?
Well, we're just now finishing the second phase of the ENCODE project, and now there will be the third phase of the ENCODE project starting. Probably it's actually about to start. And further, there are many areas in which the ENCODE project can proceed. We still only know a small fraction of how the genome works, right? Maybe 1%, maybe even less. So there are different areas. For instance, we have interrogated only a few cell types within the human genome. The human body has hundreds of different cell types, and we have interrogated only a couple dozen of cell types. So we have the need to interrogate how are the cells of the brain, difference of the cells of the muscle, difference of the cells of the bones, difference of the cells of the eyes, etc., etc.